Good afternoon, everyone. Welcome back to the afternoon session of our conference, ICISML 2022. We have an interesting workshop coming up now by CORTEVA team, who are the main sponsors for our conference. So I request Soumya Govindaraj ma'am to take over. Please introduce yourself.
Okay. I hope my voice is audible. Okay. So, good afternoon, everyone. My name is Soumya Govindaraj. I am leading a machine learning engineering team at CORTEVA. So, I've been working in CORTEVA for the last 5 years, and before that, I've been working for almost 14 years in different domains like genomics, proteomics, and bioinformatics in academia, research, and in various industries. I'm very glad to be part of this ICAI SML conference, and we are happy to be conducting this workshop here today. So today I'll be going through some of the techniques and applications of AI and ML in agriculture. So my colleagues are over here to present more in detail of various technologies in AgTech domain. So before getting started, we can quickly have a look at a video about CORTEVA. So this is us, CORTEVA AgriScience. CORTEVA AgriScience is one of the top most 5 popular seed companies in the world. And as you all know, agriculture plays a very critical role in human life and also the economics of any given country. So how did agriculture evolve over the period? So if you see in the past 100 years of history, late 1800s, agriculture was still a very family-centered profession, with farmers owning very small fields, and they were producing only for their needs, just enough to make a living. But if you see post that in 1900, that's Ag 2.0, over here, this was a revolution of industrialization, and Green Revolution started in India around 1960. And if you put in perspective, first in India with 0.25 billion people in 1920s. It became in 1960s 1+ billion. And now in 2000, it's been 1+ billion. So it has been doubling every 40 years. And in the current era, digital era, Act 3.0, this is now driven by data, and basically we are trying to build a holistic solution for farming based on the data that we obtain from multiple sources, and we perform simulations, modeling, and machine learning. And with that data-driven decisions, we are making to make agriculture and farming better for the growing population. So to give a brief about CORTEVA, so we are present globally in around 140 plus countries. We are around 21,000 plus employees in the company. And also we have many seed and crop production product. We have around 10 million plus customers, and our purpose is to provide the right mix of seeds, crop protection solutions, and digital solutions to maximize the yield and also improve profitability for farmers, also ensuring that abundant food supply is there for the growing global population. So our headquarters is in Indianapolis, and we also have other big office spaces in Johnston, Iowa, and in Hyderabad. So in CORTEVA has various divisions. We belong to one of the R&D subdivisions. So among the multiple R&D divisions that we have, seed product development is one where they focus on plant breeding and discovering hybrid variety of seeds. Crop production discovery and development works towards insecticides, herbicides, fungicides, and other crop production traits. And in biotechnology, we work towards trait development and characterization. There's also a regulatory team which takes care of product registration and stewardship. We belong to this piece where farming solution and digital focuses more on agriculture AI strategy, applied field technologies, digital solutions. We have a group of software engineers who work on application development for internal researchers. And we, myself and my colleague present over here for this workshop, we all belong to the data science and bioinformatics group, where our focus is to build AI/ML models and research around the machine learning and AI space. So as I mentioned, our focus is more towards discovery at the genetics level in all aspects, from genetics to environment, and how this seeds perform at the field, and also the management part. So the AI/ML that we are implementing and solutioning is in all these areas. And the skill set that we all have and what is required for researching in these domains is genomics, bioinformatics, protein engineering, and systems biology for the biotechnology related work, and statistics and data analytics mostly for seeds and traits, cheminformatics, data science, systems biology, and strain engineering towards the crop production discovery. And there's a lot of data science, business analytics, bioinformatics, big data, cloud computing, and the other skills that I mentioned over here that we all possess to be working in this domain. So here I've just mentioned on some of the AI/ML applications in agriculture to just give an idea of, so you might have heard these terms or worked in these methodologies, and for you to get an idea of where reinforcement learning can be applied in agriculture. So for fertilizer management, water management, resource allocation, and I've listed out, not getting into detail of each and everything, but you just for you to get a sense of where AI/ML can be applied in agriculture, you can see some of the examples over here. So here, so this picture is from National Geographic documentary called as the Next Bread Basket in West Africa. So if you see 90% of the local population, it derives its living from agriculture. On the left, you can see large farms from Chinese companies, and one on the right are small families owned by small family-owned farms. So the solutions that we aim to provide should be helping both the farmers, the big players and also the small-time farmers. So how can we use AI/ML to continue gains in agriculture efficiency? So I'll just give you an overview before my colleagues start to present everything in detail. So as you can see, data is everywhere. So taking from satellite sequences and any data on the farm. So some of the examples that I have mentioned over here. This data in agriculture is available everywhere. How do we get this data and use it for making some important decisions, and then having these images useful for us to predict what is going to come in? So that's something which we are working on by generating actionable insights from the data that we get from the farms. So here you can see, you might have seen these video clips in most of the implementations that you must have tried in your projects. So any object detection or semantic segmentation, classification of images, these are some of the things where these methodologies can be used to make data-derived decision. So in this workshop, we are going to cover how AI/ML is going to be implemented on molecular data, how do we work on proximal sensing, remote sensing using satellite imagery, and after all of these models are built, how it can be scaled up and deployed in large scale. So I'll just give a brief of who's going to present what. So Dr. Sunil Kumar, he's going to present on molecular data in using AI/ML on molecular data in agriculture. So he'll be explaining about how gene expression and any other genomic data can be utilized in the implementation of AI/ML. And then we don't have Nitin here to be presenting from 10 feet how the data can be. So this is, just imagine that you are looking at something from the molecular level and zooming out from satellite image. So this is the most tiniest molecule that you can see under the microscope, and how AI can be implemented over here. And then when you zoom out from 10 feet, how AI/ML can be implemented in agriculture. Then Chaitanyam will be explaining us about how UAV and drone images are used for some of the prediction work that we are doing from 100 feet. Sorry. And Sonal will be telling us about remote sensing work that we are doing. This is from the topmost view from 21,000 miles plus, the satellite images that we have, how we are going to work through this. And Anil will be explaining us about the implementation of all this work at production level to scale up this work in at production. So if there are any questions I can take, otherwise I think we can go ahead with the first talk by Sunil. Thanks, everyone.
Good afternoon, everyone, and thank you so much for setting up the stage. And before I start, I would like to thank Dr. Sachin and Dr. Satish for giving us the opportunity to come and present some of our work here. I think it's a great opportunity for not only for us but also for some of the students to see and learn what's happening in industry when it comes to applications of some of the recent technologies in agriculture. So if you see, the title says sequence and molecular data. Now I see that there is no crowd here which knows more about sequences. When I talk about sequences, it's mostly about the sequences of molecular data. So molecular data is nothing but things like DNA, RNA, protein. So that kind of data is highly critical, highly important for us to subject some of these methods to identify certain patterns from there that we can use in agriculture space. And there is huge amount of data which is getting generated in this space from us. So I can give you an example that every week we are able to produce 2 terabytes of data just when it comes to DNA sequences. So very basic brief of different kind of ML methodologies. Now what I've done here is I've shown only supervised and unsupervised learning. There are multiple other semi-supervised learning, reinforcement learning, but what I'm trying to show is very basic examples of 2 different types of learning, supervised and unsupervised. So I've given 2 different examples here for supervised learning. See, you have something called as classification problem. So what you do here is you basically look at the problem as a binary class problem. So you have, if you see the graph over here, what this graph represents is a DNA sequence on which certain sort of protein binds. So whether you want to predict if the protein is binding onto the sequence or not. So that is first level of classification which you can do on data set like this. Same thing we can do, and what we can do is also look at the strength of the binding onto the DNA sequence. So let's say the binding of the protein onto the DNA is highly relevant when it comes to the function of that particular piece of DNA. So we call it as gene. So for gene to function effectively, there has to be a set of proteins which bind onto the DNA. And if you can predict whether there's a protein binding there, and if it is binding, at what strength it is binding. So the strength will give us some sort of information about how strong the gene will express. And these are highly important. If you talk about the data which is for the organization, no, but public data is available. I think you have petabytes of data which is available for you to use from genomic space, genomics and proteomics. Similarly, if you see the second sort of learning here, which is unsupervised learning, so there I have given 2 different examples. One is clustering. If you see the plot here, this is a plot for a sample which is taken from cells. So just to simplify, human body has somewhere around 30 trillion cells, and every cell has somewhere around 3 to 6 billion molecules of or nucleotides of DNA within it. And each dot here represent 25,000 of such genes in this plot. Now deconvoluting this plot to identify certain areas within the body or within the tissue type is highly critical. So this is one of the examples where we can try to deconvolute the signal by looking at how different expression patterns are there. Similarly, another important thing is association. So what you do basically is you have 2 different observations, and you try to associate those 2 observations based on certain sort of either mixed model or regression kind of model to see if there is any kind of association between these 2 data types. And we can then use one of the data types to predict the effect of the second one. So like I said about the association, so here there are 2 different kinds of domains I have put in. One is genotype. So this entire space is about DNA, protein, molecular data, RNA data kind of thing. On the other side, you see a lot of phenotype data, whether you have UAV imaging, satellite data, ear photometry, which is nothing but proximal imaging. So what you have to do is you have to associate these 2 data sets to see if there is any kind of DNA data that can be used to predict some phenotypes from here. So phenotype is nothing but how a trait looks like, a specific trait. There are multiple different methods which have been applied in literature to do that. I mean, we come from agriculture, so there are 2 basic ways of doing crop improvement. One is the breeding part, and second is biotech-edited part. So this is a classical example where we use breeding. We use certain sort of data set which is kind of genotype on a small microchips. And in this case, I've shown here low, medium, high. This is nothing but the variants in the DNA which are ported onto the chip. So we can use this to screen for some of these traits. The second way is more about biotech ways of doing things that I'll come to the next slide, but this is a germplasm cloud that you can see here. So this is a plot which is generated on a data set which is being collected for almost 100 years now in the organization. So we have a seed now. We can trace the ancestry of that seed going back to almost 100 years, and how the genetic gain has evolved in that particular crop or seed of interest. Like I said, second way of doing things is through biotech ways. So idea is very simple. So you have a crop or a plant which has some sort of disease. So there is a kind of disease, AS or Asian soybean rust or anything of that sort is getting affecting the plant. Similarly, you have another trait where you have a plant which is giving you very low yield. And what the ideal scenario is, you have a plant which is highly resistant. At the same time, it has high yield. Now, from a biotech standpoint, what you need is, you need a lot of other things as well, but there are two critical things that you need. One is a trait gene. A trait gene is nothing but a piece of DNA which is present in the crop, or you bring it from outside, let's say a microbial source or fungal source. You insert into the plant, and then that plant, the specific changes in the DNA will happen for that particular plant, and it becomes resistant to that disease. Similarly for the high yield also. Now, there is a lot of discovery effort goes on to identify these genes. Similarly, there is a lot of discovery effort goes on to find mechanisms by which you can control this gene. So many of you would have heard of CRISPR-Cas. So this is one of the tools which have been currently used to edit these, create these edits. Now, my area of expertise is mostly to identify control mechanisms. How do we identify control mechanisms so that we can take the gene, we can put it in the plant, and then plant gives this kind of phenotype over here. So how it's traditionally done in last probably 2 to 3 decades is you have a trait gene of interest. Let's say this is a gene. If you put it in the plant, or if you express that in the plant, it will become resistant to a specific disease. And there is certain region upstream of the gene which is called as promoter. So don't get bogged by the biology jargons here. It's just we have a string here. There is an upstream string which is controlling that gene. Now, in case, let's say, assume that this is a gene which is expressing at very low level, and you want to increase the expression of this gene. So you take this portion which is controlling this gene, replace it with a strong promoter, and then you increase the expression of this particular gene. This is the biotech way of doing things in the most simplistic manner. Now, like I said, genomic revolution has happened. There's the amount of DNA data which is getting generated is huge. So these are some of the sequencers. Again, some of the pictures are taken from web, but we have all these sequencers. And when I say sequencer, it's nothing but it sequences the DNA in your body or plant or of organism of interest, and it produces a string. Let's say in case of maize, it is the string length is 2.3 gigabases. Now, what I will do now is I'll give you one example where I said that there are regions, or the promoter region which is upstream of the gene, how it controls the expression of a gene, and how we can use that. How we can use AI/ML methods to predict that. So what I've used is very classical methods. So just to give you a simplistic pipeline, how we go about doing this is you get the data. Now the data can then, once you get the data, you have to define what kind of problem it is. Classification, you want to predict whether the protein binds or it doesn't bind. If it binds, what is the strength of the binding using regression kind of models. So now what happens is if you talk about, let's say, plant genome, plant DNA, entire DNA of a plant, it is 2.3 gigabases long, but only 1% of that will have that control mechanism. So if I take that 1%, that is something which I have to predict. Now if I take any set of DNA within that, and I will say that there is 99% chances that it will not control, I will be right, because only 1% which is controlling. So in case getting data set which is equal in both the classes becomes very difficult. So in those cases, you have to do some sort of upscaling or upsampling or downsampling of the data set. Once that is done, you basically define some sort of accuracy metrics which will be used to say whether your model is good or bad. So in this particular example where we are trying to predict protein binding onto DNA, we use DNA sequences, we use protein sequences, some sort of structural properties of DNA, and some physiochemical properties of the DNA which are going to be used for building the model. Once you have that, there is pre-processing of data that is involved, so I'm not going to get into details of that. Once you do that, you do some sort of feature extraction, and this is something which is very critical, especially when you talk about classical machine learning, because deep learning and all kind of methods, they do it by themselves. But for classical machine learning, you need to have a lot of domain knowledge to extract these features. In this particular cases, you can see there are certain features you can take, DNA sequence features. You can look at the k-mers, small 2 bases, 3 bases, 5 bases, k-mers. You can look at some sort of motif, and motif is nothing but a sequence which is conserved across generations. You can take protein sequences. You can take additional experimental molecular data that can help you build model with a high accuracy. And once you have done that, you do feature selection. And there are multiple ways of doing feature selection. And then in this case, let's say for this particular example, I've bold made this thing here. A KMR motif, experimental molecular data. This is something which has features which are more important in discriminating between binding and non-binding. And once that you have done, then you take your data, build matrices, and start building models on top of that. You can have data set in training, testing, and validation set. And you can apply multiple different cross-validation methods and then use the testing. And again, testing depends on whether you are doing a classification problem where you can use the ROC or PR cutoff curve, or you can use regression or Spearman or Pearson correlation when you are using the regression methods. Now, so when I say pre-processing, mostly thing I'm talking about is filtering the data and identifying there's any missing data. So I'll give you an example of gene expression. So when you look at gene expression of a particular plant, we look at around 50,000 genes. Out of 50,000 genes in one sample, let's say there are 30,000 genes which are expressed. Similarly, there are multiple different classes now. There will be certain genes which doesn't express anywhere. So just filter them out, make the data small, because then it is not a burden on the computational complexity. Well, there are multiple outliers, ways to predict outliers. The classification, the clustering is something which is very, very simplistic way of looking at things. Let's say if I have gene expression data, if you see the plot which I showed in the beginning, there are certain outliers which are present in the boundary. So there are certain automated ways of doing that as well. No, this is all text data. So there's no image here. Okay, so getting more deep into this problem. So don't look at this flowchart. This I have taken from one of my previous papers, which was the work was done at Swiss Federal Institute and Swiss Institute of Bioinformatics. So what here is we generated some sort of ground truth data, and this data came from some of the experiments which are conducted by one of the bigger consortium called as ENCODE. So they have done a huge set of experiments of human samples to generate such data sets. So we took the data set, we extracted certain set of features, and then we built some model around them to see if we can predict. Now, if you see the graph here, the data which is produced by ENCODE is this blue line here. Now, what I wanted to do was I wanted to create a classification problem where I want to identify certain region, certain set in this data set which has high density of signal. So I took certain set of data set, identified which has high signal, and I call it as an enriched set. And the one which is black over here is the depleted set. So now I have two classes where I can build my classification model to see whether the protein is going to bind onto DNA or not. Now, once we do that, we have to extract certain sort of features. So this is the motif which I was talking to you about. So the motifs are kind of represented in a position weighted matrix like this. You take this matrix, you scan the DNA, and you identify, you give a basic score. So this is there is a probability of this motif to be over-represented in the sequence which you are looking at. So this is one of the features which can be used. Another which I have done here is phylogenetic conservation tree. Again, now don't go by the name. What it does basically is you have DNA sequence taken from 100 different species. And there are certain regions within the DNA which are conserved across species. So there will be something which is conserved between me, chimpanzee, gorilla, or even a banana, right? So if it is conserved across all of us, that means it has some sort of importance. Otherwise, it would have been mutated, right? So this is nothing but it shows the conservation of that particular region. So in this case, the protein binds here and you at 0 position, and you can see there is a high conservation in the enriched class. Similarly, what we have also done here is taken some other experimental data just to enhance the prediction capability of the data. Now this data can be used in a signal like this, where you can take certain enrichment in the center of the signal, or we can also do is look at the shape of the signal. So we, in a couple of years back, we had published a paper where we kind of used some sort of probabilistic partitioning to extract the shape of the signal and assign a probability value to that. Now that probability value can then be used as a feature in your model building. Now sometimes what happens is you see very weird features. Again, I shouldn't say weird features, something which is unexplainable when you look at them. Now in this particular case, like I told you, there has to be high conservation when there is a protein binding. But in this case, if you see the enriched sequence has a high frequency of DNA variants. So DNA variant is nothing but you have a string, and in that string there is one character which is changed. So that is DNA frequency. And if the region is conserved, we should not see much changes. But in this case, you can see there is high frequency of variants here compared to the depleted class. But when we look at the sequence conservation across 100 species, you see that we see a different trend. So what we wanted to do was, then we performed some sort of a fuzzy C-means clustering, where we looked at the distribution of the signal or the DNA across the samples. And you can see that most of the sequences where protein binds have low frequency, except there is a small class of 30% over here, which has high frequency of these variations. So this basically explains us why that signal was being observed. Now once that is done, what then you can do is you can build model. You can build model on one particular cell type, one particular human, one particular mouse, or those things, and you can see how that performs across different cell types. So in this case, this is a regression problem where I'm showing you the predictive accuracy was pretty high. So correlation coefficient is somewhere around 0.9. It is very good in terms of prediction. And model was built on one particular cell type and was predicted on different cell types. And these are some of the features. Again, I'm not going to go into detail because this is more biological in nature, but then you can look at certain set of features. And then when you have to predict, you can take only some of those features which are important and build your, let's say, future tools that can help you predict that. And what we also did was we kind of took that in one cell and predicted across different cell types. And this gives you which cells are similar in terms of their regulatory potential or binding potential of this protein with respect to others. So there are certain more biological hypotheses that can be derived out of this kind of data set. Again, this is one example where I'm not going to go into detail. This is work which again was done as part of Swiss Federal Institute and Swiss Institute of Bioinformatics, where what we wanted to do was, so what I told you in the previous work was mostly about looking at the data as 2D. But what we want to do is we want to look at the DNA as a whole and see how we can explain some of the variability that happens in the DNA with respect to the functioning of the DNA. So what we then did was we kind of built this map. Now, just to show you the complexity of building this map, we needed huge amount of data to generate this, and we spent around $3 million just to get this picture over here. So what it has basically is, we did a lot of experiment for a certain set of proteins, and we built what we call as CRDs, is core regulatory domains, that we also correlated along with topological associated domains, which is nothing but another method by which we can capture how regions of the DNA are interacting with each other in 3 dimensions, right? So once we did that, then what we wanted to do is we wanted to associate these regions with the either neighboring gene, which is we call as cis in nature, or in trans. Now in this you can say this particular regulatory domain is controlling 2 different genes, and this some of them will be controlled by this particular gene is controlled by 2 different regulatory domains. Now after we did that, what we wanted to see is what is the direction of this effect. So let's say if there is a genetic variant, there is a change in the string, how will that impact the functioning of the DNA? So I'm not going to explain too much of this figure. So this is built by one of my colleagues, where we applied some sort of Bayesian method to see if there is a QTL variant is nothing but DNA variant. It happens in trans or cis here. Trans is nothing but if we take 1 megabases away from the place where it has an effect. So how does that happen? In most of the cases, what we observed was if there is a genetic variant, what it will do is it will affect the regulatory domain. That regulatory domain in turn will affect another regulatory domain, and both of them will have an impact on the expression of the gene. So this is what we had done at that time. Now, another thing, this is probably the last example which I want to talk about is AlphaFold. This is nothing but a derivative of Google's DeepMind, where you can predict the structure of a protein. It is highly critical to predict the function of protein because that gives us the actual functional information. So to take an example, we are kind of making crops which are more resistant to a specific disease. So what happens is there is a protein which is responsible for that. How do we engineer that protein to get better effects? So this is where this kind of method becomes very important. And if you see both these examples here, the one in green is the one which is experimentally derived, and the one which is done in blue is computationally designed. And to get the structure of proteins is not easy. I mean, it takes years just to extract or elucidate one structure of one protein structure actually. In that sense, I think it's way too important a discovery from our work point. And again, there are the way it does is basically take MSA multiple sequence alignment. It takes a look at the structure of the proteins, how they are relating to each other, look at different domains within that, and then learn from each other and try to create the structure for the protein. I think that's what I had.
Consider from syndromes like that, genetic disorder. How to identify which syndromes for particular cause for any disorder of anything?
So I have done work actually almost for a couple of years in that space actually. Now there are multiple ways of looking at it. I think what you have to do is you have to kind of design a study where you look at the simplistic ways, look at the DNA of a person who is getting affected, you are not getting affected. Another way, I told you about the association studies, right? Those associations helps you. So what you can do is sequencing a genome is still expensive. So there was a time when the first human genome was sequenced at $3 billion, right? Now we can do it for $3,000, but it's still expensive. What you can do is you can create a small chip and you can screen hundreds of thousands of samples. So I can give you an example on one of the work which I worked when I was in Swiss Federal Institute was looking at the cardiovascular effect, genetic effect on cardiovascular diseases. So we had taken out 300,000 samples there, but unfortunately, even with that huge data set, we could only explain 3% variability of cardiovascular disease. So that is another way of applying such methodology in that domain.
So one thing is, suppose for the example, prawn production, right? Suppose you can consider suppose 6 cell or 8 cell. The number of prawns are available in that particular land. Suppose the production at in Odisha region, same production at a Punjab, right?
Yes.
So you can see in Punjab what happens, the number of prawns will be a little bit more.
Yes.
As compared to
Happens. Yes.
it will be more. So
It will happen.
Odisha. But I think all of these are different, definitely. Right?
No, what happens is when we talk about genetic gain, you have to see it from three different things. One is genotype, one is phenotype, third is environment. So environment plays a huge role. I can give you an example of cloning. When cloning first happened, there was this sheep which was cloned, Dolly. I don't know if you remember. Dolly was looking exactly like from the sheep which was from where it was taken. Second example was, nobody knows the second example was a cat which was got cloned. The cat fur was very different than from where it was cloned. So the cat was white in color, the one which got cloned was different in color. And that is because of the environment which is being, microenvironment which is there for the particular cell type. So that environment has a huge impact on how to, so what we do is, just consider this as a development process in pharma industry. It is a multi-year process, it takes around 10 years to bring one crop into production phase. And we do field testing in different geographies, in different fields, and when we plan, let's say this is a plot clip, what we do is, we take 5 different kind of crops, plants, and we place them in a statistical manner that we can kind of, their effect on each other is not getting captured in the assays which you are doing. So unbiased way of capturing that information. So that testing goes on for 2 years actually. Even with that, there will always be a certain set of differences which will happen in different areas, different geographical regions. So what we are also doing, we have a farm management system, and there is also a team from Chaitanyam and Sonam which are working on recommending certain solutions for a specific region. Let's say if you want to grow a specific corn in Haryana versus in Odisha, we'll tell you which product you should use to get best value out of that particular seed. So there's a lot of work in that sense goes on into this.
So the number of production will be high.
Yes, exactly, yes.
But we are also doing the same work in cancer disease prediction.
Mhm.
So you are in the agri molecular.
It's more or less same. So if you talk about DNA, I came from a human background actually. I worked mostly in human sciences.
My voice is more than.
So I have worked mostly in human sciences, and then I worked in mouse. From last 6 years, I'm working on plants. But in terms of DNA, in terms of molecule, in terms of data, it is more or less the same.
So I have one question. Suppose even the billion data are there. So how to find the control variable? You can superimpose the other variables. So how to identify? Because you are in the global or semi some of the matrix representation of the molecular matrix.
See, the best way of doing that is increasing the data points. The more the data points is, the more statistical significance you can get. I think there is no shortcut to that, to be honest. And if you talk about, let's say we take, something called as molecular breeding. So what it does is basically you have 30, 40, 50 markers which you have identified. And if you want to, let's say, have, there are 50 markers which are identified which is associated with yield or iron chlorosis or any of that phenotype. Now you take those and scan it in your crop of interest and see if, but to make this 50, what we have done is we have analyzed thousands of samples to identify, and these 50 will also explain only 0.4 to 0.5% variability. It will not explain the entire variability also. So that will always be there when you talk about biological samples.
Then how to identify the sample size? So there again will create a problem. Suppose you identify 1,000 samples, someone will 10,000 samples, the variation must be there.
It will always be there. What you can do is it's a trade-off, right? At some point you have to say that, okay, when I screened 300,000 samples, I have spent $10 million on that. How far we can go, right? So that there is always a trade-off there. The more the data, the better it is.
So since your data are from testicular data, so how to go to validation point of view?
Validation happens on the field for us. Yes. It's much easier for us than pharma industry who has to test it on mice, right? They will test on mouse and then on humans. For us, it's much easier in that sense. All right. Is there any other question?
Any question from audience side?
So there is a web server called as NCBI. There is a huge amount of data. So there is NCBI, Geo, Gene Expression Omnibus, and there is, I think, what is the European? ENA? So there are multiple web servers where you can get huge amount of data of any type you want actually. You want expression, you want variation, you want methylation, any kind of data you can get it from those. So if you want to know more, just reach out to me. I think probably there should be some email somewhere. Sure. All right. Thanks everyone. Now I'll hand it over to Chaitanya, who will talk things which are slightly more interesting when it comes to imaging and related stuff. All right. Thank you.
Good afternoon, everyone. My name is Chaitanyam. I'm a lead scientist at CORTEVA AgriScience. I mostly work on computer vision and machine learning problems. Today, yeah. So today I'm supposed to present proximal imaging and UAV-based imaging and other sensing, along with my colleague Nitin Kanpal, but Nitin was not able to make it today. So I'm going to present Nitin's slides as well. So the idea is Nitin would present proximal imaging and I would present UAV imaging. Proximal imaging in the sense the spatial resolution is around, Sunil talked about molecules which are micro and nanometers, right? And with proximal sensing, we call anything proximal sensing that is like 10 to 15 feet, few feet away from the object that we are looking at. And usually the spatial resolution is in millimeters. And when it comes to UAV, depends on, depending on how far your drone is and what's the camera resolution, it's usually centimeters or sub-centimeters. And we use imaging and machine learning in CORTEVA for a lot of applications. But today, this presentation is specifically for precision phenotyping because we do a lot of work with imaging in precision phenotyping along with other domains as well, but it is easier to associate machine learning and imaging with precision phenotyping. And before talking about applications, I'd like to take a couple of minutes to, let's say, tell you what is phenotyping and why phenotyping is important for CORTEVA, right? So CORTEVA at its core is a seed and crop production product company. So we sell seeds. That's where the money comes from, right? So, and we have to develop those seeds. When we develop those seeds, right, we have a lot of plant breeders that actually, let's say you want to develop a seed that gives very good disease resistance for a disease called, let's say, northern leaf blight without compromising on the yield. So what they do is they do some research and they have a lot of genetic varieties. They have like thousands of varieties. They start testing those varieties in the fields. And before taking those varieties to the field, there is a lot of research that goes from biotech point of view to arrive at those varieties. And usually, to develop one variety of seed, it takes almost 7 years. And the first 2 or 3 years, it's mostly inbred development. You basically get more and more pure inbreds, and then you cross those inbreds, and you develop hybrids. And when you start with, you're starting with 1,000 varieties. And then, after each cycle, the number of varieties that you're testing, they're coming down. right? So over the, after 7 years, you'll probably arrive with like 5 or 10 varieties, which you might want to commercialize. Okay. During this testing process, they have to collect a lot of data to select these seeds. When I'm in my first year, I have like 500 varieties. I plant those seeds in, let's say, 100 fields, right? And I'm looking for disease resistance. So I have to collect data that specifies or that identifies with the disease resistance in terms of a phenotype. And by looking at that data, I select, let's say from those 500 varieties, I select 300 varieties. Then I do more testing and I select another 100 varieties. It goes on like that until you arrive at very few varieties, right? So this entire process, let's say around 10 years back, we used to do a lot of manual work of research scientists, plant breeders, and other people. They used to go to the field, they used to look at the field, they used to take notes on their notepads, and they used to use scales to measure plant height, things like that. These are all, they take a lot of time, and it's really not efficient in terms of cost. So that's where, with the advent of technology and machine learning, we started using precision phenotyping, where we replaced this manual data collection with images, other types, imaging sensors, and other types of sensors. Okay, so first I'll talk about proximal sensing briefly. So when we talk about proximal sensing, like I mentioned, anything like, see, you can, if it's a moisture sensor, right? If it's a, you'll directly put the sensor in the field. Or if it's a camera, right, you'll probably like 2 feet away from the plant or 10 feet away from the plant, you take a picture. So there are, I mean, any of these sensors can be called as proximal sensors, but in this talk, I'll mostly focus on imaging. Yes. Moisture. There.
How to control of that variable? Because a lot of review comments are coming. So how to control? Is there any control variable to control the moisture, and how to manipulate the data?
Moisture sensors are actually a pain. So I mean, like you mentioned, right? All those challenges we have actually worked a lot with moisture sensors 3 years back, and it's actually difficult to validate the data that we're getting is right or wrong. So you use. So the point is, what we try to do is, instead of looking at the absolute data, we actually validate sensors in terms of, so for example, I have a high moisture location, like there's soil here, there's a lot of moisture content, there is soil here, there is lesser moisture content. Instead of looking at the absolute moisture, if the moisture can rank these two locations as this is high moisture and this is low moisture, as long as that kind of resolution is there, to some extent it is acceptable, but in depth about how do they, I mean, use that data reliably, I'm actually not aware, because I have not worked a lot with these sensors. I have worked with my colleagues a little bit to analyze the data, but
Then what is the use of this gyroscope?
sorry? Gyroscope, so for example, if you're collecting video data, right? Or if you're doing camera localization, if you want to know the camera exact rotation, orientation, and all.
So where to deploy the camera?
I'll talk about few applications. Yes. So, I mean, when you talk about proximal sensing, these are usually the devices that we use. The leftmost one, this is a mono camera. That's a simple GoPro camera, and we collect data using mobile devices, the cameras that are on mobile. And the next one you have is a camera with fisheye lens for a expanded view of the field view. And then you have a stereo camera which you can use to generate depth information. Then you have a 360-degree camera which you can use to do 3D reconstruction. So so as I go through the slides, I'll show an example of an image that you can collect with each of those sensors. So the first one is the mono camera. It's a simple regular cameras or digital camera. And then this is a image that we collected using a mobile camera. And the right one, if you see, a similar image, if you collect using a fisheye camera, because of the fisheye lens, you can look at expanded field of view. And then this one, so this is a stereo camera. You have two cameras. You can basically use photogrammetry techniques and estimate depth information. And then using a 360-degree camera, I mean, you have to do a lot of post-processing, but you can do some sort of 3D reconstruction. Okay, here I'm showing some applications we have built. On the top, what you can see is it's just a video stream of data that we captured in our field as we move through the field dials. And the bottom, what you see here is we have actually built an object detection. I mean, it's I think it's an open source model. We have just taken the open source model and trained an object detection model on our field video data. And you can see in real time, we were able to detect corn silks. Those are there in the small red boxes. And on the right, what you see here is, so we collect a lot of video data in our fields. Now, we want to know which of the data is actually coming from plants, which of the data is actually coming from the field dials where people walk, and which of the data is, which of the data belongs to these corn ears or corn silks. We wanted to segregate our video frames into multiple groupings to analyze the data in a easier manner. So if you see, I don't yeah. So this is just, we used a self-supervised learning algorithm to cluster our video frames from that video stream into aisles and corn ear images and other groupings of similar objects. And then this is another example. So, for example, these are pile of images. So those corn ears, they don't look good, but because they are actually they have a disease, we were actually screening those corn ears for that disease. And when we wanted to understand how severe the disease was there in that pile of corn ears, the first thing that we were supposed to do is identify which is corn ear in that image. And then once you identify, you can focus your efforts on the identified part of the image, pixels of the image, and then you can estimate your disease severity or stuff like that. So this was again an open source algorithm, and we used that algorithm to identify which are corn ears, and then on top of that, we developed additional algorithms to count the kernels in the corn ear and then disease severity, things like that. Okay, so I have, so far I have talked about some applications where we used proximal imaging. Now, so I'll talk about, we actually are doing we are using drones a lot in our patient phenotyping efforts. Actually, before even proximal imaging, we started working with drones. So 2012, I think around 2012, when we first bought our drone, and then we started collecting data and started slowly analyzing the data. And by 2014 and 15, we realized that there is a lot of value in replacing our manual data collection efforts with drone-based data collection. So we expanded our fleet, and we expanded our drone fleet to, initially it started in North America, then we expanded our drone fleet to Europe, South America, and APAC, Asia Pacific. And by 2018, right, we were actually capturing multiple precision phenotypes using drone in an automated fashion. I'll show some of those examples in the next slides. But if you look at the whole pipeline of drone data collection in terms of imaging, right? So you have a field, you fly your drone. When you fly your drone, right, let's say you have a 10 acres field. So when you fly your drone at 40 feet height, you cannot capture the entire field using a single image. So your drone flies like this. You capture one image here, then it goes to another point. You capture another image here. You fly to another point. So you actually go in a zigzag manner like this, and you capture those images. Now you have multiple images that you have captured using the drone, but you want to be able to analyze those images. You have to stitch those images and generate a single orthomosaic image of this entire field. That's what happens. That's what is happening in the second step here. we're actually stitching the image using image processing algorithms and generating a single orthomosaic. Once you stitch that image, right? So in our fields, right, the way experiments are designed is like I mentioned, we test multiple varieties. So let's say in a 10-acre field, you have 1,000 plots, and each of that plot would have, let's say, 20 to 30 plants, and within a plot, each of those 20 to 30 plants would come from the same genetic variety. It's a single experimental unit when you look at it from the experimental point of view. So the plot clip that you see is the image of a single plot of plants. Okay. Once you have this orthomosaic image, you have to generate these plot clips to be able to associate your phenotype with each plot. For that, what you need is you need a shapefile of the entire field, which actually has information about in which part of the field you have each of those plots. So that shapefile basically it's a rectangular grid. Imagine like you are cutting a cookie, right? Using a cookie cutter. It's exactly that. Your big cookie is the orthomosaic image, and your cookie cutter is the shapefile.
So what is the purpose of plot clip?
Plot clip is so.
What I'm saying, once you got the attribute, right? So number of attributes you got it. You can attributes should be you can optimize, and on the basis of optimized attribute you get the pieces.
No. This attribute is actually to generate the plot clips. So this attribution step, what we do is you have the single image for the entire field. There is a shapefile that has information about. So each of this plot clip, right? This is represented by something called a EU. It's our experimental unit. That shapefile has information with respect to which experimental unit is geographically placed at which location of the field. Shapefile information when we initially plant our varieties, right? Using a tractor or a planter, we record that information and then keep it. That is used later when we, so even if we do not do drone imaging also, shapefile information is recorded for each of our research fields because they have to know which particular variety is planted in which part of the field. Exactly.
Then when you capture the picture through drone, right?
Yes.
So all are we are assuming that all are should be same resolution, right?
Yeah, all are same resolution.
Same resolution. Otherwise the problem is occurs.
Yes.
So once you got the resolution, you plot all in a particular matrix format.
Okay, so you're saying we can clip the entire image using a standard width or standard length. It doesn't work that way because sometimes the gap between each of those plots is not always uniform.
So how to consider this all the figure?
Because that information is already embedded in the shapefile. If the shapefile gets actually, you have this single orthomosaic and that shapefile gets overlapped with that orthomosaic image to generate these plot clips because those plot clips are not uniformly placed. If the plots were uniformly placed,
So some picture must be superimposed. Then how do you move that one? That would take into by
the shapefile takes care of that, no? Right, so when we do our image analysis or machine learning, we actually do our analysis on these plot clip images and then generate whatever phenotype or trait information that is required. I'll go into some examples here. So there was a phenotype that we were collecting in soybean for maturity. So it's actually, you have to track soybean greenness over a period of time and then identify when it's going to maturity. It usually happens based on the color, when it turns brown, basically. So in this particular case, we collect sunflower images over a period of time, not sunflower, I'm sorry, soybean images over a period of time for multiple time points, then use image processing to identify the greenness within each image, and then you plot that greenness with respect to the day of the year, and then find a pattern, find a curve that fits that pattern, and extrapolate that curve to estimate when that particular plot is going to mature. Again, there are some comparisons with ground measurements to the drone-based estimations. It basically says that there is a very good correlation between ground measurements and drone measurements. In fact, in this particular use case, we have proven that drones have delivered better data than humans. Similarly, there's a sunflower flowering phenotype that we have developed, where you have to, I don't know if you can see, there are small yellow flowers here. Our objective is to identify, count the number of flowers that you have in the image, and then again track them over a period of time and estimate when that sunflower flowering plot is going to flower. So if you look at the image here, that is basically a result of estimation of, counting of sunflowers from the previous images, and then you basically plot it against time, you fit a model, and then extrapolate it to estimate the sunflower flowering date. Similarly, we score plots for disease severity using images. It's again, you have 3 different plants of varying levels of severity of disease, and we do simple semantic segmentation kind of algorithms to identify which are healthy plant pixels and which are unhealthy plant pixels, and then based on that we score those plants. And then similarly, again, another use case for soybean, where we were, it's a nutrient deficiency condition. We were using image processing techniques on drone collected images to basically score those plants on a scale of 1 to 9, 9 being a very healthy plant and 1 being a very sick plant. Okay, again, we have done some validations, and we have actually established that between visual data collection and drone-based data collection, there is high level of correlations. And so far, the applications I have been showing, they all were dependent on some sort of color or hue. But in this particular case, we wanted to collect plant height. Plant height is even more challenging to collect manually, because there's a lot of subjectivity involved, and everyone has their own style of collecting data there. So we actually collect overlapping drone imagery, simple RGB imagery. Then we use photogrammetry techniques to generate digital elevation map surfaces, and then we use the DEM maps to estimate plant height. Okay, so I think that's all. If there are any questions.
How will you here? Slides? I'll have to unlock it. No, here. Oh, no, this it's HDMI होता है ना? HDMI लगा देते हैं बस. Power charged है पूरा. I'll unlock it. सौम्या सेंड क्या? Is it working? Hi everyone. I'm Sonal from CORTEVA. I am a remote sensing data scientist. I've been in CORTEVA for past 11 and a half years. Since four and a half years, I've been working in remote sensing space. And as Sunil was, Soumya was talking about that how we are starting from 10 nanometer and going up to thousands and thousands of feet. It is no easy feat, and to integrate different scales together to produce a product and enrich the lives of farmers, which directly enrich our lives and the generations to come, is, I think, is a very, very big feature. So, until my slides come up, sorry, there's a technical glitch. Where is he? Okay. So I'm just going to talk about and give you guys an idea about what satellite data sets are. So, and what satellites are basically. So, just like Chaitanya mentioned, that there are drones that are flying 100 feet above us. There are satellites that are going in orbit around the Earth, basically giving us eyes to look at the Earth in its entirety. And it also tells us about at what scale we can analyze the data. So there are multiple open source satellites that are available. There are obviously which are very, very high resolution. They are expensive to use. We work with both depending upon the kind of project that we have taken up. There are two different classes of satellite which are widely and vaguely classified. One of them are optical satellites, which basically works during the daytime when you have sunlight, and they don't take images when it's dark. And there are other types of satellites which are called as synthetic aperture radar satellites. Those are basically microwave satellites. It's not coming. Okay. So optical satellites, so how we perceive the color green in the plants is basically they absorb certain wavelengths reflected. Okay, great. How do I change this? Oh, here. Right. So I was talking about there are two different kinds of satellites. One are optical and one is SAR. SAR, I'm going to give a history about the SAR, the working with SAR in CORTEVA. Because we are not using, I mean, we do use optical as well, but specifically for India, our main season is Kharif season, which basically starts from May and ends in November. And the two most important months for analyzing data, which is July and August, which are the peak season, are usually covered by clouds. So you can see if there are clouds, optical data will not be utilized, which is very unfortunate because it can give a lot of information. But SAR data, which is basically a microwave data, which can give a lot of information about the geometry and the structure of, let's say, a plant, like plant height, its biomass, its width, its greenness. Sorry. So that is why I'm going to focus more on SAR, and maybe if there is time permits, we can also talk about optical data. So the radar, initially it was, we were calling them radar because these are microwave systems. They can understand and sense the amplitude and the phase of the reflected microwave signals, like you can see in the animation. The satellite radars are, they will bounce back to the satellite and provide you the data depending upon the structure and the height of whatever object of interest that is there. So you can see PlanetScope is one of the very expensive, I should tell you, is an optical satellite, and because of the cloud, because of the cloud, you can't really see the image properly, you can't see the fields clearly, but SAR satellite on the same day, you can actually pretty much visualize it properly, right? So this data is far better to build a machine learning algorithm and identify, let's say, for example, we are looking at corn crops. So this data would be much, much better than if I, use PlanetScope on a very heavy cloudy day. This is SAR principle. So this is basically what it's doing is it's sensing that red color object on there. and then the antenna footprint will capture the combination. So basically, it's not just one satellite that are there. So let's take an example, Sentinel-1 satellite. It's constellation of multiple satellites in the sky, and it takes multiple acquisitions of the same area. And when it does, you get a really, really high resolution satellite. So it's 10 meter. Right now, there are multiple satellites. It starts from 1 meter and goes up to 250 meters. So this is like, Sentinel-1 is like a moderate resolution satellite of 10 to 20 meters. So there are a lot of different, like you can classify a land of interest into different objects. There are So there are buildings, there are trees, there is bare soil, there may be some water bodies, there may be forest areas. So SAR can actually trace back with, let's say there are phenomena called double bound scattering, volume scattering, which can tell you about the biomass and the width of the tree or a plant, the rough surfaces, bare soil, and when it comes back, like when it bounces back, it brings a lot of information about the object. I'm gonna skip this, it's the same thing. So this is just giving you the history of when it started. The legacy satellite started from 1978, which was mostly basically used for American military. And now it's like we have NISAR, which is again a SAR satellite. In 2021, it was released. We still don't have hands on the data yet because it's still yet to launch its, in its open source entirety. And then there is Biomass, which it says 2021, but it hasn't launched as of yet. So there are different bands at which the satellite captures the data. L band is the most penetrative one. It can penetrate deep into the soil. X band, not so much. It can cover only the top canopy of the field or crop. And C band is something that we mostly used because it has been in literature as well, and for our experience in the industry also says that C band is something that has been very, very crucial for agriculture monitoring work. Different resolutions, partial coverages are available for the satellite data. Some of them are 10 meters to 20 meters to 30 to 250 meters as well. If you are doing like North America fields, they are very, very big. Brazil fields are very, very big versus the fields in India are very small. Smallholder fields are available. So in that, you cannot use something which is 250 meter because that wouldn't make sense. Some of them revisit. So imagine a satellite going in its orbit and then coming back. So that is its revisit or temporal frequency, which can be, for Sentinel-1, it's 6 to 12 days. For some other satellites, it may be 3 or 4 days. So how quickly it comes back to the same position, that's the revisit frequency of the satellite. And that is very, very important because when you're doing like disease monitoring or health monitoring of the field, temporal frequency should be as high as possible. You wouldn't want to wait that long for the next data to come up. I'm gonna skip some of these quickly because of interest in time and come back to the work that we have published in, this was one of the work that we published in Google conference. This is Geo for Good Summit. This was the work we did for our business team, basically. We provided them with fall armyworm focus. Fall armyworm is a pest which is highly menacing in corn crops. And it is not native to India. It was introduced in 2018, and the first time farmers saw was in Karnataka. And since then, a huge yield losses has happened because of fall armyworm. And we really have a very good chemistry in CORTEVA to combat fall armyworm, but the only problem is we needed to help the business team so that they can position their products, in such a way that farmers get the most benefit out of it, because you can apply that chemistry only within a window of 20 to 45 days. So we wanted to forecast the fall armyworm risk before the season actually even starts, so that there is ample time for our representatives also to go and help the farmers on the field so that they can apply the chemistry and mitigate the risks of fall armyworm on the field. So how we did that? We used satellite data We have the ground data information which is coming from our field scientists. They do a lot of survey in collaboration with farmers. And for corn, the major business areas for CORTEVA includes Karnataka, Maharashtra, Bihar, AP, Telangana, some areas in Uttar Pradesh and some areas in Haryana. So it is a wide, it is a way larger area than compared to a 10 nanometer and a 10 feet where you can't really fly drones in such large areas and understand what is happening. So what comes into picture is a satellite data where we can analyze these crops at a much, much larger scale. And then this data, the ground truth data, gets augmented with satellite information, with weather data, because weather data plays a very, very critical role in infestation, pest infestation, its development and its spread. And using all the data sets, we are building cat boost algorithm, and then it giving us good accuracy because we are doing it at such larger scale. So for us, even 80 to 85% accuracy is very, very good. And that is also very helpful for the business. So we are, with that much accuracy, we've built the algorithm to be able to forecast it well 15 days in advance before the season starts. And then once the season starts, every 15-day interval, we are helping the business team with the forecasts. So yeah, that's what we did. So I think we don't have time, right? It's okay. Thank you. So here we are, I mean, you are asking in terms of the model that we have developed. So we are using cat boost model. It's a tree-based decision algorithm. So it takes, the good thing about cat boost, it also is very helpful when you have categorical features in your data set. In this case, we had. So it's helping us to. Nominal meaning? Nominal, you mean numerical data? No, you can. You can. It doesn't have to necessarily be a certain kind of this data set or that. We tried, we have. I wouldn't say that we haven't. My first choice was ARIMA. But then, since it's very difficult to have satellite at exact interval of time. So we didn't have, like a proper cycle of time series data. So that's why we went ahead with cat boost, which learned the pattern in a much easier way. At least we are able to interpret what it is doing. But with ARIMA, it was very difficult.
Thank you very much, ma'am. Thank you, Team CORTEVA, for giving your valuable sessions. Actually, ma'am said only 2 presentations, so because of, okay. This is the last one. Yeah, okay, ma'am. Thank you, sir. Please continue, sir.
Hello. Yeah. Thank you all. This is Anil. I'm working as a machine learning solution architect in CORTEVA. My task mostly related to the deployment of the models. Whatever they are presented, it is all needs to be scaled and productionized to make it use for the end users. So our task resides on that part. So when you are going through the machine learning models, whatever they are building, it was for the specific sample data. It's not across the whole data. So we have to iterate it multiple times to make the model happen. After that, the deployment cycle will happen. If you could see the cycle here, it will start from data preparation and exploration of data analysis. Then you have to go for reviewing the data, train the model, and see whether it is going by the performance standards or not, like whether you are getting better accuracy or not. If it is not there, you have to develop it again. You have to prepare the data again. So the cycle will go on. Once we are finding out that particular model is fine and we can go for the deployment, we will go. The next cycle will start from there. That is the deployment cycle. Sorry. That is the deployment part where you need to start whatever model it is there, you have to deploy it and make it available for the outside world. And by using those deployed models, you have to infer the data. Like for whatever data they checked, it is all the sample data or test data. So for the real-time data, after deployment only you can test through the inference part. Once you are going through the inference of some certain data, like 1 or 2 years of data, you get to know there is a data drift or model drift. So if there is anything like that, you have to monitor those results and go back to the development mode and give a new version of the model. So this cycle will happen all the time because the data will change, the environment will change, so it will always happen. So for our CORTEVA things, we have, this is the cycle for all our image pipelines. The first part is the image acquisition. You saw the multiple modes of image acquisition. What we are doing, satellite images we are acquiring, the normal camera images, and the molecular data as well. But for the image, we are only doing through the cameras, satellites, and everything. So those acquired images, we need to put it into one of the repository. For that, before going into the repository itself, we have to annotate those images. Usually for the training the model, you need some of the test labeled data. Like you already, for the training the model, you have to get to know the images which has already labeled, whether it is a field or corner, anything like that. For that, we have to do the step of annotation. It can be performed through automation or it can be performed through manual intervention, but you have to annotate the data and push back the data into the image repository. So that image repository should contain all the images, like raw images and the annotated images along with that. And once the images are available, we will do the pre-processing step and also the image analytics development, like the model development and which model we have to use. Those things actually will happen in the image analytics development part. And later, they will do the training and evaluation part. Once they are coming up with training and evaluation, and they will come up with the model as the outcome for that phase, and we have to do the production deployment after that. If you are looking at the different stages, we are using different tools for each stage. For the model training and image analytics, we are using TensorFlow, OpenCV, Keras, SQL, and a lot of available platforms they are using to make the model happen. And after that, for the production deployment stage, we are using the Kubernetes. And in the Kubernetes was built on top of AWS. So for our pipelines, we mostly use for those two. Yeah, if you are looking at the pipeline components, what I have explained earlier, I'm just showing with the examples here. Image capturing, you could see the different kind of images we are capturing in the first box. And also the image annotation. For the image annotation, we are using the different algorithms and the tools. Supervisely is the automation tool. You can do the automation and also manual labeling part also in the supervisely. And label me is the labeling diff, open source label tool. You could use it to annotate the data. And in a similar ways, we could use an NVIDIA digits label tool. All these are available tools for the image annotation. For image processing and analytics development, as I shown earlier, it was the different frameworks available right now, PyTorch, TensorFlow, Keras, and everything. And for the same part of models we have developed, we could use the different deployment methods. The available methods for the production deployment right now is TensorFlow serving, TF serving, PyTorch serving, Flask applications. You have to build your model from the scratch and you can deploy it using Flask. And there are TensorFlow Lite, Argo. These kind of methods you could use to productionize your models. So when we are going to the productionize our models, I will classify those productionizing things in 3 different forms. One is the batch prediction, and second one is the on-demand prediction. Third one is the embedded prediction. Like if you are going with a batch prediction, how the batch prediction will work is, you have the lot of image data. We will capture all the seasonal data into the one image repository. In our case, it was media pillar. Whenever there is a new image data, we want to apply the whole, whatever models applied till now, we got till now, we have to apply that in that particular images and make the prediction happen, and we want the inferred data on top of it to make it into available for the end users to check whether the performance is good or not. So for that, we will do the batch prediction kind of pipelines. We will build the pipelines using Argo workflow, where we could define the steps called getting downloaded the data, and after downloading, you have the different services explored with the existing models. So we call that particular model based on the requirement of that project, and we will do the prediction happen, and we will push back the data again into the media pillar. And apart from this on-demand prediction service. This is the REST kind of implementation. If there is a model, we want to expose that model as a REST service. So whenever you hit that service with the required parameters, you will get the inferred data. Like if you are giving image as input, you will get the annotated image as an output. So that kind of deployment we did using on-demand prediction. For this on-demand prediction, we are using Sheldon Core as a standard protocol for our CORTEVA side, but there are multiple tools available for that in the market. Like Flask, this is the very basic implementation of our model prediction. And TensorFlow serving, Amazon SageMaker, PyTorch, all these are platform-specific servers. Like you could deploy the models which are built on top of these frameworks. But Sheldon Core is the framework where you can deploy any kind of platform output as a model using Sheldon Core. That's why we went with the Sheldon Core. And Kubeflow also you can implement for these kind of requirements. But we are in the process of implementing that part. And the third part is the embedded models. Embedded models are nothing but you don't have to deploy it anywhere. You have to deploy it in your app or in your edge device. Anywhere you are, whatever hardware you are using, you have to deploy the model inside that hardware. So for that kind of setup, there are the examples like DeepLens. DeepLens is the Amazon's product, where you have the pre-trained models inside the device itself. So whenever you take the picture, you will get the annotated data directly. So in a similar manner, we have different kind of tools already implemented services are there. One of them is Granular Business, HAI, Plantix. Usually, we use some of our projects. We got explored for this DeepLens, and Granular Business also one of our implementation. And for our deployment, as I said, we are using Sheldon Core. So we are using Sheldon Core on top of Kubernetes. So I just want to give a brief introduction about the Kubernetes, but as the time is ticking, I will just explain the main services here. Inside the Kubernetes cluster, there is a client-server architecture actually here. One is the master node, remaining are the worker nodes. You could make the all communication will happen through the API server. So whenever you want to communicate through the Kubernetes cluster, you have to hit the API server. API server will give the response according to based on the request. So there is a cloud manager, cloud control management, which will take the details of the cloud resources, and it will make it available, the resources available. Like if you want a new pod to spin up, it will check if you are deploying your Kubernetes cluster, any of the cloud provider like AWS or Azure. It will communicate with that cloud provider and get to know the details about the resource specification. And it will make sure it will be available for all requirement. Like if you are trying to spin up 10 pods, it will request that many resources from the cloud, and it will make it available for you. And etcd and ingress and everything, these are all related to your communication. It will maintain your state of all worker nodes, like how many resources are available, how many nodes are there, how many resources are available. If you are coming up with new request, etcd will decide where you want to deploy that particular pod. Whether node 1 is free, node 2 is free, it will check the resource table and it will spin up the pod in that particular pod, in that particular node. And if you, I'm just explaining here the sample web application, how you could deploy in the Kubernetes. There are 3 kinds of deployments we need to do. One is the deployment, second one is the service, third one is the ingress. So one is the deployment where you could configure all your application details and you could give the details about the image which you want to deploy. And you could mention what kind of port you want to expose, and you can give the name of the deployment, which you can use it for the next mapping purpose. And whenever you are deploying things, it will not be available for the outside world. You can only access through that particular cluster nodes only, but you want to see all these results in the outside world. So we have to configure the service for that particular deployment. That mapping will happen through this app name. We will configure inside the service configuration. So from the service configuration also, you can't see the, you can access through the IP, but usually what you will see the outside world is the URLs, like google.com. You want to hit that URL and get the response. So for that purpose, you have to configure the ingress, and ingress will be configured on top of service. So whenever there is a request, it will come to the ingress, and it will coming back to service, and from the service, it will hit the deployment. And this is the cell and core setup, how you are going to do any new model. There are 2 types of models where you are directly giving a prediction function definition and write the wrapper and make it as you can directly use that model for inference. You will hit that API and you will run the inference, and you will see the model binary, model binary working, and you will see the output of that particular model. But there are some models where you need to pre-process the image. Like if you are giving input as an image, you want to convert them as a NP array, and you have to do some pre-processing, like you have to filter out some of the regions from the image, then only you can apply the model. So all those pre-processing stuff you could install and set up through this complex cell and core inference graph. This graph we have to define. Once we define the graph, you have to, whenever you are hitting the API, it will perform all the pre-processing steps, and later it will run through the multiple models available for the particular input. So whenever which model is performing better, it will command those results and getting back that result as an output. Yeah, this is the model serving, how the model serving is happening inside the cell and core. It was taking it from the central repository, or you could write the wrappers as well. For our purpose, we are mostly writing the wrappers, Python wrappers, to make it available for the deployment purpose. And once you are deploying and everything is running as expected, you have to find it out where the model starts dipping their performance and where the data drift is coming. For these parts, actually, the monitoring and logging is very important. So for those purposes, we are using these Thanos, Grafana, Cortex. These are available tools in the market. So for our purpose, we are using Grafana to make the watch on the model performance. Like whenever you are hitting the model, we are just logging out what are the performance for that particular model and what are the intermediate steps it got performed, and what was the metrics, like how many epochs we configured and how much performance you are receiving for that particular prediction. We are monitoring everything into the logs, and we will make it as a dashboards to see whether the accepted performance is coming through that particular inferences or not. Over the time, we will get to know if there are any dip in the performance, if there are any change in the data, it will automatically dip the performance. So at that time, we will configure alerts from the Grafana, and you will get to know there are performance dip. So in that way, we were again notifying the data scientists to make it use a separate model or make it release the next version of the model. And these are some of the references I have used to prepare this presentation. And if you have any questions, you can ask me.
So here a few couple of stories are there. The model validation, right? Data validation model which are two different things. Right?
Model model validation is when you are developing a model. Like after training, you will validate the model. Yes.
Suppose I validate the data before I apply the model, right? Is there required model validation or not?
We are not doing any model validation. We are just configuring to get the performance stats. That's it. We are not doing any validation.
Rely the output of the model will be accurately perfect. How to rely on it?
We will get to know the performance matrix. In the performance matrix, we will get the every probability of the prediction. Like if you are solving some classification problem, you don't directly get whether it is cat or dog. You will get the probability of that, whether it is cat or dog. For cat, it was 90%.
Basis of that probability percentage, we can find the prior quality or posted quality. We can find that the model is perfectly wrong, right? My question is, how to test the model? Model is accurately correct. Suppose you can call the robustness, right? This model is particularly robust or not? How to?
That we can't know automatically. You have to.
We can put a different variety of data. If the output is coming, then we can show that if accuracy level will reach or satisfy, then we can call model is valid.
So, for an example, so you use a lot of our machine learning models to generate results.
Yeah.
So, let's say we have put a model in production. We are not collecting ground data. Okay, so you do not have ground data to compare your model against anyone. In that case, what you do is, once you have your phenotypic data, breeders do a lot of statistics, like nativity calculations, or is the genetic signal that they are expecting is coming or not. They do. So they actually look for those flags. If they find something is wrong, then they actually go back and look at the data, the model generated data, and see something that they did not expect from that model. Let's say they come back to us, they ask us something. We have given this input to the model, and it has generated this.
No, I understood. Suppose a set of data is there, we can find the various type of model. So which model is the better accuracy maximum to other model? That in this way, we can compare that. This is our model is accurate. My question is something little different. Suppose X model is there, right? Suppose you develop X model, Madam develop X1 model, Sir develop Y1 model. Same data set. Are you thinking that all the model have not same performance, maybe different performance?
Yes.
Suppose any two model out of n model are having exactly same accuracy. So which model will be?
Which is the most computationally efficient?
And efficient means what? Efficient means
Which has most.
No, that's what I'm asking. Now, suppose n1 and n2 two model or n1 and n2 two model out of n model.
Okay.
So out of n1 and n2, how you claim that n1 is more both the same performance? How to same performance?
If the performance is same,
How to say?
you look at how much computation cost it takes to run each of those models. Let's say you're talking about M1 and M2 model.
Yeah.
So M1 model and M2 model you're getting same accuracy.
Yeah.
So whatever metric you're looking at, then from our point of view, if it takes 5 seconds to do the inference using M1 model.
Okay, time to predict the concept.
10 seconds. That means M2 is costly.
Yeah.
And also generalization factor also we will take care. Like how much generalized data we have. That also we will consider the Thank you.
Thank you very much, Team CORTEVA. Now I request Dr. Princey ma'am to speak about a few words regarding the. Okay, Nidhi ma'am. Now I request Nidhi ma'am to address the gathering.
A very great thanks to all the CORTEVA people who have enlightened us with their knowledge, their learnings and the findings which they have done recently in the machine learning field. We all wish you a very good luck for the near future and expect that we'll be getting such type of knowledge and experiments, new findings from you all. Thank you so much.
Thank you very much, Dr. Nidhi ma'am. Now I request Dr. Sachi sir to felicitate the CORTEVA team.
So I like to thanks to all the entire team. They have done a very good work. Of course, it's related to my work. I'm working in the cancer disease analysis of molecular human life. So they are doing the in agriculture. So in crop production, or we can call your crop recommendation system. So it will now the agriculture is a very major thing. Now government of India also required many type of data analysis of subject in this particular contest. So those the young students are sitting, my students are sitting. So you want to do the internship, right? So they are paying the paid internship, right? So you can contact Dr. Sunil and Dr. Sweta, you can contact. So they are providing very good internship, and the international guy, I think Mustafa and other thing, you can contact. So they are providing a paid internship, even as a PhD MTech students, right? PhD and MTech students. So definitely they will come forward and help to you. So end of the day, we have to do some fruitful work so we can benefit for the society. So that is our human value. So whatever you do the work, do for the some at least from the society. So we can do, so you can selfish no problem.